Hi, everyone. Welcome to Naturally Immune for Life. Happy Mother's Day. It's a beautiful sunny day. I'm in my relaxed clothes and I'm going to zip through this because it's take 3 and I forgot my sugars. And now I have to get back to my article. Excuse me. But this week's article is on autophagy and how autophagy keeps your cells healthy and how insulin can shut it down. So this is the bridge between autophagy and metabolic syndrome. And understanding the science behind cellular recycling, its history, and the hormonal roadblocks that can disrupt it. And insulin is one of those. So autophagy, derived from the Greek words auto, which means self, and phagy, which means eating, or phage, which means eat, is a fundamental cellular process where cells degrade and recycle their own components. This mechanism is crucial for maintaining cellular homeostasis, especially during periods of stress or nutrient deprivation. By removing damaged organelles and proteins, autophagy prevents cellular dysfunction and contributes to overall health. So if you did not have a functional autophagic system, you would accumulate up to 2 tons of cellular debris in your lifetime. So the historical perspective. The concept of autophagy was first introduced in the 1960s following the discovery of lysosomes by Christian Dedeuve. Electron microscopy revealed the presence of double membrane vesicles, later termed autophagosomes, which engulf cellular components for degradation. However, the molecular mechanisms remained elusive for several decades. A significant breakthrough occurred in the early 1990s and a Nobel Prize when Yoshinori Osumi utilized yeast models to identify genes essential for autophagy. His work elucidated the steps involving autophagosome formation and highlighted the conservation of autophagy-related genes across species. And I believe he learned all this through looking at trees. These discoveries laid the foundation for understanding autophagy's role in human health and disease. So the mechanisms of autophagy primarily occurs through 3 pathways: macroautophagy, microautophagy, and chaperone-mediated autophagy. Macroautophagy is the most extensively studied form, involving the formation of autophagosomes that engulf cytoplasmic material and fuse with lysosomes for degradation. Microautophagy. In this process, the lysosome directly engulfs cytoplasmic components through invagination of its membrane. Chaperone-mediated autophagy is a selective process that involves chaperone proteins recognizing specific substrates and directing them to lysosomes for degradation. Autophagy is regulated by various signaling pathways, with the mammalian target of rapamycin, mTOR, playing a central inhibitory role. Under nutrient-rich conditions, mTOR activity suppresses autophagy, whereas nutrient deprivation inhibits mTOR, thereby activating autophagy. So, in that one sentence, in those two sentences, I explained something extremely difficult. Which suppresses what and which activates what. So insulin's role in autophagy regulation. Insulin, a hormone crucial for glucose metabolism, also significantly influences autophagy. Under normal conditions, insulin suppresses autophagy through the activation of the P13K AKT mTOR signaling pathway. When insulin binds to its receptor, it activates phosphoninositide 3 kinase P13K. Is that one? I'm not sure if that's a one or an I. Leading to activation, I think it's an I, of AKT. Activated AKT then stimulates mTOR, which inhibits autophagy. I think it's a P. I. Okay. Moreover, insulin affects autophagy. I'm going to have to look that up now because it's bothering me. Might just be my font choice. Moreover, insulin affects autophagy by modulating transcription factors. For instance, insulin inhibits the activity of FOXO1, a transcription factor that promotes the expression of autophagy-related genes such as BPS34, ATG12, and GABA-RAPL1. By inhibiting FOXO1, insulin reduces the transcription of these genes, thereby suppressing autophagy. I do think it's an I now. Moreover, implications in insulin resistance and disease. In conditions, it's an I. In conditions like insulin resistance and hyperinsulinemia, commonly reserved in type 2 diabetes, the suppression of autophagy becomes more pronounced. Studies have shown that in insulin-resistant states, autophagy activity is diminished, leading to the accumulation of damaged proteins and organelles. This accumulation contributes to cellular dysfunction and exacerbates metabolic disorders. Furthermore, the impaired autophagic process in insulin-resistant individuals affects various tissues, including the liver and skeletal muscle, leading to disrupted energy homeostasis and increased susceptibility to diseases such as non-alcoholic fatty liver disease and muscle atrophy. So in conclusion, autophagy is a fundamental cellular process essential for maintaining cellular integrity and function. The regulation of autophagy by insulin underscores the intricate balance between nutrient signaling and cellular maintenance mechanisms. Disruptions in this balance, as seen in insulin resistance, highlight the importance of understanding autophagy's role in health and disease. Continued research in this area holds promise for developing therapeutic strategies targeting autophagy to treat metabolic disorders and other related diseases. So, health tips. can you do? Health tips to naturally induce autophagy. Practice intermittent fasting, allowing your body to go without food for 14 to 18 hours can activate cellular repair and cleanup processes. Reduce processed food intake, in turn lowering insulin levels. Lower insulin levels are a key trigger for autophagy. And that is because they have almost zero fiber, and whatever nutrients they have are usually fake. So it's not good for nutrition. If it's not good for nutrition, then it's going to affect autophagy. Avoid excess sugar. High sugar intake leads to frequent insulin spikes, which directly suppress autophagy by activating the mTOR pathway. Exercise regularly. Both aerobic and resistance training stimulate autophagy, especially in muscle and liver tissue. And if you can't do aerobics or resistance, swimming is a good choice. Or you can try a vibration platform. Sleep well. Poor sleep increases cortisol and insulin resistance, both of which can block autophagy. Avoid constant snacking. Eating every 2 to 3 hours keeps insulin elevated and prevents your body from switching into repair mode. So if you want to learn more, that sounds like Jason Fung, who is a very good teacher in trying to make complicated things more simple. So autophagy and gut health in IBD. Autophagy plays a key role in keeping the gut healthy, especially in protecting it from harmful microbes in the intestine. Types of autophagy, like macroautophagy, help trigger immune responses and fight infections. But when this process doesn't work properly, it can lead to chronic inflammation, as seen in conditions like inflammatory bowel disease. Obviously, I'm beginning to lose my voice. Nutraceuticals, functional foods, prebiotics, probiotics, polyunsaturated fatty acids, amino acids, that's some amino acids, and polyphenols and other gut-associated adjuvants can reestablish gut tolerance by altering the gut immune system and/or via modulation of intestinal microbiota. Some amino acids, fatty acids, short chain and omega-3 fatty acids, conjugated linoleic acids, and oleic acid, and probiotics, bifido, Saccharomyces, and lactobacillus can restore the intestinal barrier, supporting gut barrier integrity and function. While research acknowledges many adverse effects due to leaky gut syndrome, they do not support it as a sole factor as the cause of IBD, leaning more toward a dysfunctional autophagic system. And you can learn more in the link in that infographic. So, okay, everyone, I will be placing this in the metabolic health in the feature section, as well as it'll be, well, that's pretty much it. I'll be placing it there so you can learn about the bridge between autophagy and metabolic syndromes. Okay. So happy Mother's Day. Have a great day, a great week, and thank you so much for your time. And don't forget to like, share, and subscribe. I thank you very much. Bye, everyone.